After studying this module, you shall be able to know the composition of a nucleotide, chemical structure of nucleotide, what type of bonds exist in the nucleotide, how Watson and Crick proposed the model of double helix DNA, and what are the features of double helix DNA. We will start with a brief introduction of DNA, and later we will discuss about its detailed structure and its features. DNA structure. It is known to everyone that DNA plays a vital role in living organisms. We know that deer only give birth to little deer, tiger to tigers, dog to dogs, and so on for every type of living creature. But why is this so? The answer lies in a molecule called deoxyribonucleic acid or DNA, which contains the biological instructions that make each species unique. DNA, along with the instructions it contains, is passed from adult organisms to their offsprings during reproduction. DNA is the chemical name for the molecule that carries the genetic instructions in all living things. Now, let us know where is DNA found. In organisms called eukaryotes, DNA is found inside a special area of the cell called the nucleus, because the cell is very small and because organisms have many DNA molecules per cell. Each DNA molecule must be tightly packaged. This packaged form of DNA is called a chromosome. During DNA replication, unwinds so it can be copied. At other time, in their cell cycle, DNA also unwinds so that its instructions can be used to make proteins and for other biological processes. But during cell division, DNA is in its compact chromosome form to enable transfer of new cells. Researchers refer to DNA found in the cell's nucleus as nuclear DNA. An organism's complete set of nuclear DNA is called its genome. Besides the DNA located in the nucleus, humans and other complex organisms also have a small amount of DNA in the cell structures known as mitochondria. Mitochondria generate the energy the cell needs to function properly. In sexual reproduction, organisms inherit half of their nuclear DNA from the male parent and half from the female parent. However, organisms inherit their entire mitochondrial DNA from the female parent. This occurs because only the egg cells and not the sperms keep their mitochondria during fertilization. Now, let us discuss that what does DNA do and how are DNA sequences used to make proteins. DNA contains the instructions needed for an organism to develop, survive, and reproduce. To carry out these functions, DNA sequences must be converted into messages that can be used to produce proteins, which are the complex molecules that do most of the work in our bodies. Each DNA sequence that contains instructions to make a protein is known as a gene. The size of the gene may vary greatly, ranging from about 1,000 bases to 1 million bases in humans. Genes only make about 1% of the DNA sequence. DNA sequences outside this 1% are involved in regulating when, how, and how much of the protein is made. DNA's instructions are used to make proteins in a two-step process. First, the enzymes read the information in a DNA molecule and transcribe it into an intermediary molecule called the messenger ribonucleic acid or mRNA. Next, the information is contained in the mRNA molecule, is translated into the language of amino acids, which are the building blocks of proteins. The language tells the cell's protein-making machinery the precise order in which to link the amino acids to produce a specific protein. This is a major task because there are 20 types of amino acids, which can be placed in many different orders to form a variety of proteins. Now, let us discuss DNA structure. DNA is made up of the chemical building blocks called nucleotides. These building blocks are made of three parts: a phosphate group, a sugar group, and one of the four types of nitrogen bases. To form a strand of DNA, nucleotides are linked into chains with the phosphate and the sugar groups alternating. The four types of nitrogen bases found in nucleotides are adenine, represented by A, thymine, represented by T, guanine, represented by G, and cytosine, represented by C. The order or sequence of these bases determines what biological instructions are contained in a strand of DNA. For example, the sequence ATC GTT might instruct for blue eyes, while ATC GCT might instruct for brown. The complete instruction book or genome for a human contains about 3 billion bases and 20,000 genes on 23 pairs of chromosomes. Further, the nitrogenous bases fall into two categories: purines and pyrimidines. The two purine bases are adenine and guanine, whereas the pyrimidine base present in DNA is thymine and cytosine. The nucleotide further consists of phosphate and deoxyribose sugar, which forms the backbone of the structure of DNA. The sugar is called as 2'-deoxyribose because there is no hydroxyl group at 2' position. Now, we will study about the nucleotide composition of DNA. The purine is a double ring structure. The base is attached to 2'-deoxyribose by the removal of a molecule or water between the hydroxyl group on the 1' carbon of the sugar and base by forming the glycosidic bond. Likewise, the phosphate is attached to 2'-deoxyribose by removing water molecule from phosphate and hydroxyl on the 5' carbon. Next, we move on to the structure of the polynucleotide chain. The nitrogenous base linked to pentose sugar through the glycosidic linkage in order to form a nucleoside. The phosphate group which is a component of DNA, links to 5' hydroxyl group of nucleoside to form nucleotide through phosphodiester linkage. The two or more nucleotide link together through 3' to 5' phosphodiester linkage to form a polynucleotide. DNA is a polynucleotide. So, in this manner, a long chain of polynucleotide constitutes a DNA. Now, we'll study about Friedrich Mischer. DNA, which is an acidic substance, was first identified by Friedrich Mischer in 1869. Due to the technical limitation of the structure of DNA, remained unavailable for a long time. Also, he named the nucleic substance as nuclein. Although few people realized it in 1869, and it was a landmark year in genetic research because it was the year in which Swiss physiological chemist Friedrich Mischer first identified what he called nuclein inside the nuclei of human white blood cells. The term nuclein was later changed to nucleic acid and eventually to deoxyribonucleic acid or DNA. Mischer's plan was to isolate and characterize not the nuclein, which nobody at that time realized existed, but instead the protein components of leukocyte, white blood cells. Mischer thus made arrangements for a local surgical clinic to send him used pus-coated patient bandages. Once he received the bandages, he planned to wash them, filter out the leukocyte, and extract and identify the various proteins within the white blood cells. But when he came across a substance from the cell nuclei that had chemical properties unlike any protein, including a much higher phosphorus content and resistance to proteolysis, that is, protein digestion, Mischer realized that he had discovered a new substance. Sensing the importance of his findings, Mischer wrote, "It seems probable to me that a whole family of such slightly varying phosphorus-containing substances will appear as a group of nucleins equivalent to proteins." More than 50 years passed before the significance of Mischer's discovery of nucleic acids was widely appreciated by the scientific community. For instance, in a 1971 essay on the history of nucleic acid research, Erwin Chargaff noted that in a 1961 historical account of 19th century science, Charles Darwin was mentioned 31 times, Thomas Huxley 14 times, but Mischer not even once. This omission is all the more remarkable given that, as Chargaff also noted, Mischer's discovery of nucleic acids was unique among the discoveries of the year. Major cellular components, that is, the proteins, lipids, polysaccharides, and nucleic acids, in that it could be dated precisely to one man, one place, and one date. Now, we'll study about Watson and Crick. Morris Wilkins proposed the first clear X-ray images of DNA in 1950 to 1951. In 1953, Rosalind Franklin gave Wilkins a high-quality image of B form of DNA, which Wilkins showed this to Watson. This image, together with the major contributions by colleagues in Cambridge and the data obtained by Franklin, helped Watson and Crick to propose a double helix model of DNA. For this work, Watson and Crick, along with Wilkins, were awarded Nobel Prize for Medicine in 1962. Now, moving on to X-ray diffraction. It is used for determining the atomic and the molecular structure of a crystal in which the atoms cause a beam of X-rays to diffract into many specific directions. The structure of DNA and many other biological molecules was revealed by this method. Here, in this method, a beam of X-ray from the source X-ray tube is allowed to pass through the lead sheets, where the beam of X-ray falls on the crystal, where the diffraction pattern can be seen in the photographic plate. Many biological macromolecules will not or cannot crystallize. However, an important group of fibrous macromolecules such as DNA or many of the components of the cytoskeleton form oriented fibers in which the axes of the long polymeric structures are parallel to each other. Often, as in the case of the muscle fibers, the orientation is intrinsic. Sometimes, the long molecules can be induced to form oriented fibers by pulling them from a gel with tweezers, sometimes by flowing a gel through a capillary tube, or even by subjecting them to intense magnetic fields. The experimental setup is rather simple. The oriented fiber is placed in a collimated X-ray beam at right angles to the beam, and the fiber diffraction pattern is recorded on a film placed a few centimeters away from the fiber. Fibers show helical symmetry rather than the three-dimensional symmetry taken on by the crystals. By analyzing the diffraction from oriented fibers, one can deduce the helical symmetry of the molecule, and in favorable cases, one can deduce the structure. In general, this is done by constructing a model of the fiber as in DNA and then calculating the expected diffraction pattern. By comparing the calculated and observed diffraction patterns, one eventually arrives at a better model. Maurice Wilkins, the nuclear physicist, entered the race for DNA based on a stroke of luck. Early in his career there, he happened to attend a conference where a biochemist gave away samples of high-quality DNA. Wilkins was lucky enough to get a sample, though it might not have seemed that impressive at that time. It was so slimy and gooey that he later described it as just like snot. Nevertheless, because it contained long intact DNA molecules which were hard to come by at the time, the slippery sample would turn out to be critical in uncovering the clues to DNA's structure. Raymond Guzlin, a PhD student in Wilkins' lab, suggested looking at in DNA with a new observational technique called the X-ray diffraction. X-ray diffraction, developed in the first half of the 20th century, was one of the new technologies that made solving the structure of DNA possible. The technique works on the crystal, a kind of molecule with a regular repeating structure. When X-rays are aimed through a sample, they are bent or diffracted in different directions depending on the locations of the film. Because the X-rays must travel through many layers of atoms, it's important that the atoms always occur in the same crystalline arrangement. If they don't, the X-rays are bent into overlapping patterns or leaving the results a fuzzy, indistinct blur. However, if the structure has a repeating arrangement of atoms, they leave a pattern of sharp, clear spots. Different structures scatter the X-rays into different characteristic patterns. Even the scientists couldn't directly observe the atoms within the crystal. They could work backward from X-ray diffraction patterns to reconstruct the three-dimensional structure that produced the scattering. This works a little like trying to figure out how tall a person is by looking at his or her shadow. Depending on the angle of the sun, the shadow might be longer or shorter. But if you could compare many pictures of their shadow at different times of a day, you eventually be able to figure out how tall they were. Similarly, scientists compare shadows or X-ray diffraction patterns cast by a crystal to determine the arrangement of atoms within it. Although the DNA didn't look very crystalline, Guzlin wanted to know DNA's structure might be too irregular to produce a clear, well-defined X-ray pattern. But as it turned out, the sample was sticky and stringy because it was made up of lots of long, thin molecules of intact crystalline DNA. Over the summer of 1950, Wilkins and Guzlin's patterns showed that DNA did have a regular structure, which meant that X-ray diffraction would be a critical tool in solving the structure. The patterns even suggested that what the basic structure might be. Despite a few confusing blurry spots, the images hinted that DNA might come in the form of a twisted spiral, better known as a helix, though it was still not clear how the phosphates, sugars and bases were arrayed within that helix. Now, moving on to the salient characteristics of double helix DNA. The salient characteristics of DNA structure are as the DNA is made up of two polynucleotide chains where its bone is constituted by the sugar and phosphate. DNA structure comprises of two chains which run antiparallel to each other. These chains are called as strands. One strand runs in 3' to 5' direction, while the other runs from 5' to 3', that is in opposite direction. The pairing takes place between the base pairs. A purine will always pair with the pyrimidine, that is adenine, which is a purine, and thymine, that is pyrimidine, always pairs up with each other by two hydrogen bonds. Guanine, a purine, always pairs with cytosine, which is a pyrimidine, by 3 hydrogen bonds. The schematic diagram has been shown here, which shows the pairing of all the 4 nitrogenous bases with the formation of 2 and 3 hydrogen bonds. The two chains of DNA molecule are coiled in a right-handed manner. The pitch of the helix is 3.4 nanometers and each turn of the helix contains 10 base pairs. Here in the figure, we can see that the plane of one base pair stacks over the other in double helical structure of DNA. Now, we will study about the forensic significance. Genes are the blueprints of life. Genes control everything from hair color to blood sugar by telling cells which proteins to make, how much, when and where. Genes exist in most cells. Inside a cell is a long strand of the chemical DNA, that is deoxyribonucleic acid. A DNA sequence is a specific lineup of chemical base pairs along its strand. The part of DNA that determines what proteins to produce and when is called a gene. DNA testing has become an increasingly popular method of identification and research. The applications of DNA testing or DNA fingerprinting within forensic science is often what most people think or when they hear the phrase. It is also frequently used for other benefits like wildlife studies, paternity testing, body identification, and in studies pertaining to human dispersion. While most of the aspects of DNA are identical in samples from all human beings, concentrating on identifying patterns called microsatellites reveal qualities specific and unique to the individual. During the early stages of the science, a DNA test was performed using an analysis called restriction fragment length polymorphism or RFLP. Because this process was extremely time consuming and required a great deal of DNA, new methods like polymerase chain reaction and amplified fragment length polymorphism have been employed. The benefits of DNA testing are ample. In 1987, Colin Pitchfork became the first criminal to be caught as a result of DNA testing. Now, let us summarize all that we have studied in this module. DNA is made of nitrogen bases. The first nitrogen bases present in DNA is purine and pyrimidine. The two purine bases are adenine and guanine. The two pyrimidine bases are thymine and cytosine. Next component present in DNA is phosphate group and deoxyribose sugar. The nitrogen bases is linked to pentose sugar through glycosidic linkage in order to form nucleoside. Phosphate group, which is a component of DNA, links to 5-hydroxyl group of nucleoside to form nucleotide through phosphoester. The linkage between two or more nucleotide is done through 3' to 5' phosphodiester bond to form a polynucleotide. A well-known scientist, Friedrich Mischer, in 1869 named nucleic substance as nuclein. The data produced by Rosalind Franklin and Maurice Wilkins, Watson and Crick for the very first time in 1953 produced the double helical model of DNA by X-ray diffraction. The significant features of DNA structure are the DNA comprises of two polynucleotide chain where its bone is created by sugar and phosphate. The two chains of DNA run antiparallel to each other. The pairing takes place between the base pairs. A purine always pairs with pyrimidine. That is, adenine always pairs with thymine by 2 hydrogen bonds. Guanine always pairs with cytosine by 3 hydrogen bonds. The two chains of DNA molecule are coiled in a right-handed manner. The pitch of the helix is 3.4 nanometers and each turn of the helix contains 10 base pairs. One base pair's plane aligns over the other base pair's plane in a double helix.